Alright, so we're going to be talking about homeostasis. So homeostasis is a process that our body goes through to maintain an internal stable environment when there are external environmental changes happening. So one example of that is with temperature. So our body temperature stays about the same or relatively the same. It stays within a normal range. These aren't the exact numbers, but pretty close for a normal range within our body. And so the temperature outside could be 102 or it could be negative 20 degrees, in our internal environment temperature is going to stay the same. It's going to stay around this set point range. And so if it goes out of that normal range, it can become a bad internal environment, and it can even become lethal for us, something that we can't live in. And so how our body maintains this homeostasis is through feedback loops, different control center signaling and messaging that goes throughout our bodies. And so there's different kinds of feedback loops that happen. There's positive and negative. So here's an example of a negative feedback loop that happens in our body. So say we just eat a meal, our blood glucose is gonna rise right after eating. So then our pancreas is going to release insulin into our body. And from there, the insulin stimulates glucose intake, and so then the blood glucose goes back down to normal. So the key with negative feedback loops is that the response reverses the original change that is happening. Whereas with positive feedback loops, there aren't a whole lot of situations where this is a good thing, but one is with childbirth. So as the baby pushes against the cervix, it triggers oxytocin release, and the oxytocin strengthens the contractions that are happening, and so then more oxytocin is released, and this continues until the baby is delivered. And so with positive feedback loops, that means that the response is amplifying the initial change. So with this one, the oxytocin, it's released once, and then it continues to be released throughout the duration of the cycle and through the change that's happening. And so then from here, this is kind of the control center where all the signaling happens, but the signaling and the ions need to move between cells, and so that's where protein-mediated transport comes in. So there's a couple different kinds of transport that happens. So there's diffusion channels and carrier-mediated diffusion. Both of these are happening without the use of extra energy. It is moving with the concentration gradient. So this one, it is moving completely on its own between membrane and just through the gradient. With carrier-mediated diffusion, it has to bind to another molecule, and through the molecule, that's when it's kind of like a little bus driver, a little chauffeur that takes it through the membrane. And then with active transport, active protein-mediated transport, there's also two kinds. There's primary and secondary. So with primary transport, it is moving with the use of additional ATP, energy, that kind of stuff. Secondary is moving without, but the key with active is that it's moving against the concentration gradient. Both of these are moving with the concentration gradient, whereas this one is moving against. And so that's how the signaling and ions move between membranes. But water follows a little bit of a different pattern. That pattern, that cycle, is called osmosis. Simply it means just the movement of water. But the water follows wherever the higher concentration is. It follows the higher volume of molecules. And just to try and gain equilibrium and try and equal out the concentration of molecules. Alright, so now we've got some more terms with osmosis. So osmolarity and molarity sound pretty similar, but they're two different things. So molarity measures just the solute concentration, just generally the solute concentration. Whereas osmolarity, it measures the solute concentration, the particle concentration, but how it influences water movement, so related to osmosis more. And so with these, we have some cells, and some IV bags. And so in each of these cells, it has 300 about milliosmoles of concentration, and then the IV bags, ones with the cute little triangles, is 0.9% of NaCl, and then the little circles is 5% of dextrose. And so with these ones, they both contain, and both of these are equal to the 300 milliosmoles. So both of these solutions are going to be isoosmotic because they have the same concentration in both the cell and the IV bag. And then this one is it's hyperosmotic because once we combine the NaCl and the dextrose, there's a higher concentration living in the IV bag than there is in the cell. And so hyper means that there's extra, hypo means that there's too little. And so this one's going to be hyperosmotic. And so then we're going to look at these little pictures. This has to do with tonicity. So tonicity measures a solution's ability to cause a cell to gain or lose water. So whichever one has a higher concentration is going to have the water follow it in order to maintain that equilibrium. And so it changes with whether the cells are able to penetrate or if they're non-penetrating to the membrane. So that goes back to the protein-mediated transport. If they're able to go through the membrane on their own, or if they need assistance. And so this solution of NaCl, it is non-penetrating, so it's not going to go into the cell from the IV bag. It's just going to stay on the outside. So that's what this picture is right here. So that's once the cell and the IV bag are combined. The NaCl is going to stay on the outside, and then the cell's going to stay. All the cells are going to stay on the inside. And so this one is going to be isotonic because there's the same concentration on the inside and the outside. With dextrose is penetrating, so it will go into the cell, and so this one is going to become hypotonic because the dextrose is going to start going into the cell because it's able to merge through and penetrate the membrane. It doesn't stay separated, and so then this solution on the outside, the IV bag, is not going to have as high of a concentration as it does on the inside, making it hypotonic. The other cool thing with dextrose is that once it enters the cell, it metabolizes, and so it pretty much just disappears. And so we see down here, once we have this hyperosmotic solution, once it's combined, the NaCl is going to remain on the outside, right? And the dextrose is going to merge to the inside and become immediately metabolized. And so it's going to remain isotonic because if we look up here, it's going to become pretty much the same as this diagram right here because the dextrose just goes in and it immediately metabolizes and pretty much disappears. And so this one is an isotonic solution.